Hi, everyone. Thank you for joining our workshop, Introduction to Proteomics using the R software programming language. This is a new chapter, so we are very happy to start this new chapter, Tunis R user group. The goal of this chapter is democratizing data science and bioinformatics and letting people from Tunisia, Tunis, or everywhere in the world from low and middle income country also to learn bioinformatics and to learn something from, it's like our workshops. So thank you for joining. I'm Hediat Nani. I'm postdoc at Pasteur Institute of Tunis. I'm also co-organizer at Our Ladies Tunis and Tunis R user group. And before starting this workshop, I want to present our speaker. So thank you, Laurent, for accepting our invitation. Thank you very much. We are really happy to have you today. And so Dr. Laurent Gatto is an associate professor of bioinformatics at the Douve Institute at UC Louvain in Belgium. His research group focuses on the development and application of statistical learning for the analysis and comprehension of large-scale biological data. Since 2010, he has focused on the processing and analysis of mass spectrometry-based quantitative proteomics data in general, and spatial proteomics data in particular. The development and publication of scientific software is an integral part of his work, as reflected by his numerous contributions to the Bioconductor project. Laurent is an avid open research advocate, making his research output openly available. He's a Software Sustainability Institute Fellow and a Data and Software Carpentry Instructor and NASAP Bio Ambassador. So thank you, Laurent, very much for being with us today and for accepting, for us to learn proteomics and to introduce us to this amazing work of proteomics.
Okay.
The screen is yours.
Thank you very much. So the plan today is to give an introduction of a couple of packages that can be used to explore and understand mass spectrometry data. And so these are bioconductor packages. And they, well, as kind of any R package, expect from you some familiarity with, not necessarily with mass spectrometry. I'll try to give you enough insight on how the technology works, but I'm assuming that you have at least some familiarity with R. So let me start by sharing my screen. Okay. And so thank you very much for this introduction. My goal here is really to, as Edia mentioned, the goal is to help you get started, or maybe you're already an expert in one aspect or another of this topic, what I'm going to present. But by all means, please feel free to interrupt me anytime to ask any questions, any clarifications that you might want. And so let me start by giving a small introduction here. The package that I would like to present today are all part of an initiative, the R for Mass Spectrometry initiative. And so here's the webpage. It's rformasspectrometry.org. And the idea of this R for mass spectrometry initiative is to, let me immediately click here on the package link, is to provide a set of packages that work together coherently to be able to tackle all or specific aspects of mass spectrometry assays. And I'll be focusing, or my main area of expertise is proteomics, mass spectrometry-based proteomics. But these packages can also be used if you're interested in metabolomics. So really anything that, any data that stems from mass spectrometry. And so the goal here of all these packages, and there are many more now, is to work together and act as building blocks to provide a kind of coherent set of classes and functions to analyze these data. And the package I'll be focusing mostly on today is the Spectra package that defines a certain set of classes of computational objects that allow you to load raw mass spectrometry data into R and then handle, filter, process, and visualize these data. And the idea here is to provide a way to do this efficiently. And even though I'll be demonstrating this on small data on a single file, because I'm working on my laptop here, all this will scale up to hundreds of thousands of files through some computational tricks that I will hopefully get time to discuss a little bit later on. So the mass spectrometry-based Alpha Mass Spectrometry Initiative is the kind of umbrella organization where we develop these set of packages. And when I say we, it's mostly 3 people. So there's myself, and then there is Sebastian Gibb and Johannes Heiner. The 3 of us have been collaborating already for many, many years now. And they are also the main contributors to another package that some of you might have heard of, which is called MSMBase. Okay, but so let me maybe get started with, and so I saw that this link was posted in the chat. So rformassspectrometry.github.io/docs. And this is a kind of a book-like or kind of book format documentation or tutorial that describe various aspects of mass spectrometry-based proteomics data processing. Every package, so let me maybe go back here. Every package comes with its own web page, including lots of documentation, and of course the reference manuals for all the functions. So there is much more that you can read about the individual packages, but I will roughly be following the content of that main tutorial today. And the first thing I would like to do is to explain how mass spectrometry works, because I'm absolutely convinced that if you want to be good in data science or bioinformatics in particular, it's extremely crucial to understand the data that you have at hand. I don't think you can treat a matrix of quantitative data of, let's say, transcriptomics data or proteomics data in exactly the same way. Each of these different types of data will have their specific properties. And I really believe that it's important to understand where the data comes from, what the data looks like, to be able to make informed decision when it comes to its analysis and its understanding. So let me start by providing an introduction as to how do mass spectrometers work. And let me maybe start from here on. So this kind of black box here is my representation of a mass spectrometer. Okay. A mass spectrometer is composed of 3 parts. First, the source, and then the analyzer, and then the detector. But for any reasonably complex biological sample, it's not going to be possible to take that sample and immediately inject it in the mass spectrometer. That's not possible because that sample will be way too complex. In terms of proteomics, we will have way too many peptides, because we study peptides, even though it's called proteomics, but the material that is analyzed on the mass spectrometer, that is acquired in the mass spectrometer, are peptides. There are way too many peptides in a normal biological sample to be analyzed by direct injection into this mass spectrometer. So the first step is always a step of separation. That is done using mostly liquid chromatography. And so let me move back here at this previous figure. This figure shows you what happens when you do chromatography. And so a chromatographic column is like a very, very long tube on which the peptides will stick, depending on their chemical properties. And then slowly, the pH of that column is going to change. And when it changes, the column will release more peptides. So in the beginning, and the time that it takes for these peptides to get out of the column is called the retention time. So in the beginning, you have all the peptides that stick on the column, and then slowly over time, you have more and more peptides that are released from the column. And the time they stay in the column is called the retention time. It's written here, right? So that's the retention time. And this figure shows on the x-axis this retention time in seconds. And then on the y-axis, we have the total signal that was measured by the mass spectrometer. Right, so this kind of figure can only be produced at the end, after all this has happened. But at the moment, we focus on the very first step. And so we see that at the very beginning, here we have no or very little peptides, and then suddenly you have peptides that come out of the column, that enter the mass spectrometer, that are analyzed by the mass spectrometer, and then we have a decreasing amount of peptides, and what is measured is the ion current, a decreasing amount of peptides and decreasing amount of signal. And this here at the end is just some dirt that is flushed out of the column. Right, so if we were to inject the whole sample directly into the mass spectrometer, we would essentially have all this signal that is condensed in one injection, and that's not possible. So the first step is a separation of these peptides. This is done using chromatography. And so the first dimension of our data set is going to be the retention time here. And the mass spectrometer will be working, will be acquiring new data constantly along this retention time. So at every, sorry about that, at every second here, and multiple times during a second, the mass spectrometer will be working and will be acquiring some data. And so what does it mean, acquiring some data? Well, that is illustrated. That's what happens inside this box. So acquiring data first means transforming these peptides that are liquid when they come out of the chromatography column and transform it into a charged gas, so into positively charged ions. And that's what happens in the source. So you might have heard of ESI, electrospray ionization. That's most of the mass spectrometers, high throughput mass spectrometry uses electrospray ionization. You might also heard of MALDI. That's the kind of another type of source that ionizes the peptides with a laser. So peptides are liquid when they come out of the chromatography, and the first thing that happens is that they are ions, positively charged ions, are created in the source. And why do these peptides need to be charged? It's because the only thing that the mass spectrometer does inside the main piece here, the analyzer, is separating charged molecules, charged particles based on their mass to charge ratio. So every molecule that enters the mass spectrometer gets ionized in the source. It has a specific mass to charge, m over z. It's written here. So m stands for the mass of that peptide, and z is charge. It might be singly charged, doubly charged, triply charged. While all these peptides that enter the mass spectrometer are going to be separated in the analyzer based on the different m over z values. And after separation, these ions are going to hit the detector that is going to measure how much of these different ions that are characterized by an m over z value entered the mass spectrometer at a very specific time along the retention time. And so what is recorded? Well, something like this. That's a spectrum. And here on the x-axis, you see the m over z value. So all these ions here entered the mass spectrometer at a very specific retention time. And here at the top, we see after 21 minutes and 3 seconds, they all entered the source of the mass spectrometer together. And then they all got separated based on their mass to charge. And then they hit a detector that measured the intensity, that gathered a measure that is proportional to the amount of peptides that we had in that entered the mass spectrometer. And so if you want, this is the unit of data that is acquired in a mass spectrometer. These are scans. And these are kind of two-dimensional objects. These are matrices that have hundreds of rows, and they have two columns. One column that represents the m over z value of that particular ion and the intensity at which it was measured. Any questions so far? Okay, so here's a little illustration, a little video that illustrates what I just said. So now, at the beginning, here we have all the ions that are characterized by a specific m over z value. So the small dots that you see here have small m over z values, and the large dots have large m over z values. And what you see here, the separation of these ions based on their m over z value is what happens in the analyzer. In the beginning, they're all together, and now they get separated based on their mass to charge. And then they are measured by a detector that produces this figure, which is a spectrum. It tells us how much analyte that is currently characterized only by its m over z value and its retention time were acquired. So we can visualize these data in 3 dimensions. And here it is. So along this dimension here, we have the retention time. Okay, for example, here 21.1, 21.15, 21.30, and so on. And in a continuous way, we have scans or spectra that are measured, that are a set of data, a set of ions that are characterized by their m over z values, and that then are measured by the detector. So you can kind of think of mass spectrometry data as this cube of data spectra that are acquired constantly over time. But if you think about it, the only thing that we know, and let me move back here. So these peaks that you see here are peptides. But the only thing we know about these peptides is that they have a specific mass to charge. So for example, if we look at the red one here, right, this peak here that is highlighted with the red dotted line has an M over Z value of 460.79. And I know this because it's written out here. And we also know that that peptide that has an M over Z value of 460.79 came out at a retention time of 21 minutes and 3 seconds. But so that's not very much. I mean, that's not very much information that we have, and that's certainly not enough to be able to characterize the peptide, to be able to figure out what peptide this was, what sequence it had. And without that information, we can't relate it back to what protein it came from. And that's what we're interested in, right? We want to do proteomics. We want to measure the proteins that were present and in what quantities in our respective samples of interest. So what happens to be able to characterize that peptide here more specifically? And that's what's illustrated still here on my figure. So all these peaks here, the individual peaks, are called precursor peaks. That's the peak of the peptide. Well, what's going to happen, and that happens automatically, very, very quickly in the mass spectrometer, is that a specific M over Z region here is going to be selected around the M over Z value of interest. So each of these dotted lines, so I have one red dotted line, one orange dotted line, and 8 other blue dotted lines, these highlight the M over Z value of one of these precursor peaks that correspond to a peptide. Well, these M over Z values are now selected automatically by the mass spectrometer. And everything that is just to the left, so the selection only keeps the M over Z value of interest with a little bit of room on the left and on the right, and everything else is discarded. So that for the second round of mass spectrometry, so this is called MS1, we will only keep ions that come from that peak of interest. And then next step is to break that peptide into pieces. And that's later how the identification works. And maybe we'll have time to discuss that in more detail later. So that peptide gets broken into pieces, into fragments that themselves are charged. And then these fragments now are going to get analyzed by the analyzer. That means they're going to get separated and then measured by the detector. And this is what we see here on the right-hand side. So the figure here at the top, where we see here M over Z of 460.79, was produced by selecting the peak here that is highlighted by a red dotted line. All the rest is discarded. And then the molecules here at 460.79 are now broken into pieces into a specific part of the mass spectrometer that is called, and this process has different names depending on the technical aspects, such as CID, collision-induced dissociation, or well, let me find other ones. I'm not sure anymore. Here we see we have ETD, HCD, so kind of with different energies to break to break that these molecules here into pieces, which are that now themselves separated in the analyzer to produce another scan.
Laurent, please, is it possible to make the screen bigger? Because
Is
thank you.
this?
Yeah, it's quite better.
Oh, that maybe. Oh, okay, I think that's better.
Thank you.
And so the fragments of this red peak are broken into pieces. Also, that the ions here are broken into pieces, and the fragments of this one are now separated to produce another scan, but this one is called MS2 scan. Here we have an MS1 scan where we separate the peptides, and then individual peptides are selected, broken into pieces to produce another scan, but this time it's a second level scan, and so it's called an MS2 or an MSMS scan. All these peaks are the result of breaking the initial peptide into pieces. The scan that you see here is also an MS2 scan that originates from a precursor that has values that had a precursor charge or precursor M over Z of 488.8, which is exactly the one that you see here in orange. And this happens automatically for a certain number of scans that are selected for each MS1 scan.
We have a question.
Yes.
So Mariam is asking, could you inform us about the criteria that should be taken into consideration to choose the right column for MS for a better characterization of the peptide?
So I'm certainly not an expert in chromatography, but typically there is a trade-off between the length of the column and how much time you want to invest to acquire data. So if you have a very complex sample, then let me come back here. If you have a very complex sample, and then you would want to have a very long column so that you have a very long chromatography time to separate peptides as much as possible. But that might then produce runs that last for 2 hours. On the other hand, if you have many, many samples to process, then you can't always wait for 2 hours until you acquire the data for one sample. And then you might want to have shorter columns and a higher flow rate to be able to kind of acquire your data more quickly. So that will very much depend on the complexity of your sample. That's all I can say in terms of how to choose or how to optimize your experiments in terms of chromatography. And how to know that, but of course that's something that you will know not only by running your chromatography, but running your whole mass spectrometry experiment. And then you will be able to check how many peptides do you identify. You might even look back at the raw data and to kind of assess how good your separation was. But yeah, so it's a trade-off between sample complexity and how much time it takes, or how much time are you happy to wait for data acquisition. I hope I answered your question. So as a summary here, separation of our data outside of the mass spectrometer using liquid chromatography. You could also have gas chromatography if you do metabolomics data acquisition. A source that ionizes our peptides, and then a first round of analysis that produces MS1 scan. And then some of the peptides here are going to be selected, broken into pieces, and that's called precursor ion dissociation. And then the fragmented ions are themselves going to be analyzed and then hit the detector. to produce now an MS2 scan. And so I showed you this three-dimensional figure where we have here a collection on the MS1 scans, but in between these MS1 scans, as shown here, we have a series of MS2 scans. And each MS2 scan is the result of selecting and reanalyzing one specific peak in MS1. Okay. And so let me check another figure. Yeah, this one. Can I increase it? Okay. So with this knowledge that we have about how mass spectrometry works, it's now important to give you an idea of how does mass spectrometry data look like when we think about it in terms of computational data for the Spectra infrastructure. And so when we think about mass spectrometry data, on the one hand side, we have a large table, and it's a data frame, or you can think of it as a large data frame.
Laurent, sorry, they said that the picture is not clear.
Yes, but I'm afraid there is nothing I can do. Is it better like this?
Is it better like this?
Okay, you don't need to be able to read what's on the figure. So it's on the point here is to explain the concept. There is absolutely no value in being able to read the numbers here. That's not important at all. I will explain what some of these columns mean, and then we will actually see, we will load some data, and we actually are going to see in R what these data actually contain. But yeah, maybe I'm better off with this one. It's a little bit sharper. But there is absolutely no need to be able to read the detail. So mass spectrometry data, on one hand side here, on the right, on this figure, imagine mass spectrometry data as one big table, where each row of the table corresponds to one scan. And so for each scan, we have some information. We have an index. Was it the first one, the second one, the thousandth one, the 100,000th one in our set of acquisitions? We have information about its MS level. Was it an MS1 level? Was it an MS2 level, MS1, MS2, and so on? We have its retention time, and all sorts of additional annotation about these individual scans. And all the scans are shown as a data frame aligned along the rows, and then the columns are variables about these individual scans. And in addition to that, for each of these scans here, we have the actual raw data, which corresponds to a scan. So each of these kind of plots here is exactly something like this. Let me increase the size. Okay, so this is a scan, and on the other figure here, I have represented a collection of scans, lots of scans. Again, we don't need to see exactly what the values are here. But so each of these also is annotated by a scan, which is a matrix with two columns, one that provides the M over Z values, and the second matrix provides the intensities. So once I'll start to do some demonstrations, think of the data as something like this. A big data frame with as many rows as we have spectra, And for each spectra, we have a collection of variables of annotations. And we also have, for each scan, a matrix that gives us the information about the actual raw data, which are the M/Z values and the intensities. Okay, and so what I would like to do with you today is to load such a data set and show how we can access and how we can visualize the different parts of a mass spectrometry data set. Basically, I would like to demonstrate with an actual raw mass spectrometry data what I have just explained here. Extract the different parts, produce these kind of figures to really bring together the theoretical understanding, which is what I have explained now, and then the practical understanding to be able to actually handle, manipulate these data in R. And I will be basing everything I do on this tutorial, and the link was already shared in the chat. And so, and I'm going to do this in R. So what you see here on the right-hand side is a normal R console. So it's currently completely empty. And then here on the left-hand side, I will be writing some code to demonstrate some of these data manipulations in R. Okay. And so, as I mentioned, the package that we are going to use is called Spectra. And so here I'm going to load the package Spectra with the function library and then the name of the package. Now, if you don't have Spectra yet, you will need to install it. And this can be done with a function called install from the package BioC manager. And you can install Spectra like install, I don't know if you see these pop-ups showing up. Sorry about that. Oh, you know what? Let me delete this. So I assume, or maybe some of you will have installed Spectra, but if not, you can install Spectra by calling the function install from the package BioC manager. And this is the general way to install any package from Bioconductor. And to be able to install the package BioC manager before being able to install the Bioconductor package, you can do this with the function, with the classic install packages BioC manager. So this line of code will install BioC manager from CRAN. So CRAN is the central repository for R packages. And once you have BioC manager, you can then use its function install to now install packages that don't live on CRAN, but packages that live on as part of the Bioconductor project that has its own package repository. Are people here familiar with Bioconductor? Could you maybe reply in the chat with a +1 or -1 if you know, if you're already familiar with, okay, GG3, yes. Okay, +1. Okay, so I see a -1. So I will very briefly talk about Bioconductor because it is an extremely important project if you do bioinformatics in R. Okay, and so here I see lots of -1s and no's. So this is the web page of Bioconductor. It's bioconductor.org. And as you can see here in this About section, Bioconductor provides tools for the analysis and comprehension of high-throughput genomic data. But it's not limited in genomic as in transcriptomics. It's much more general than that, tools to analyze high-throughput biology data. And there are many, many packages. So somebody mentioned ggtree. So let's see if I search ggtree here. And indeed, so here in the search bar, I search a package of interest. And here I get the link. And each Bioconductor package has these landing pages with general information about the package of interest. Okay, so ggtree is one of them. The one that we are going to see today is called Spectra. So here it is. So Bioconductor is a collection of R packages that are specific to the analysis of high-throughput biology, whether transcriptomics, other genomics, methylation data, proteomics, or metabolomics data. And these packages get the extra round of review and are meant really to work together and are dedicated to high-throughput biology data. In R, of course. So that's kind of a biology-related or bioinformatics-specific CRAN-like repository of R packages. And so to install these Bioconductor packages, you will need to use this specific function, because this function knows that it needs to go and look for the package on Bioconductor and install any Bioconductor dependencies if needed. So assuming you have BioC Manager or you have installed BioC Manager, you can now install Spectra and then load the Spectra package. Okay. And so the first thing I would like to do with you is to create an object that contains mass spectrometry. And so I told you that the way we see mass spectrometry is, first of all, with as a big data frame. So we're going to create a data frame manually, just to kind of give us a better understanding of how these data look like to be able to manipulate them. So we're going to create a data frame with one column that says MS level, right? A level of mass spectrometry, either 1 or 2. Maybe retention time. So this is a small data set that only contains two scans. And so let's say the retention time, I don't know, is 10.1, 10 second, and then I don't know, 24 second. It doesn't matter what I type here, as long as I'll always provide 2 pieces of information. And here I'm going to limit myself to providing information for 2 scans. One piece of information is an MS level, and the other one is the retention time. Okay, and here it is. So I have a data frame with 2 rows. So let me store this in a variable called SPD. Okay, so this now corresponds to the first part of our mass spectrometry data. It's a simple data frame. But if you remember, and let me get back to that figure here. If you remember, the way we are going to think about mass spectrometry data is a data frame. In this case, it's a tiny data frame. It has 2 rows and 2 columns. In reality, they're, of course, much bigger. But in addition to that, we have raw data being M over Z values and intensities. So let's create M over Z values and intensities for our data set. And so these are created as lists. So I'm going to avoid typing too much here. I'm going to simply do some copy and pasting from the tutorial. And I'm going to explain to you exactly what I do here. So I create a list, okay, here with the list function. And that list contains two elements. The first element that's going to contain my M over Z values. So my first scan will have 4 peaks. One that has an M over Z value of 100, the next one 103, 104, and 106. That's for my first element. The second one will also have M over Z values, but in this case only 3, 45, 120, and 190. And so what I'm going to do is that I'm going to add a new column to that data frame that I'm going to call MZ. So let me execute this here, and let's have a look at how the data frame looks. Now I have my original MS level and attention time, and now I have a new column. In this case, it's a list column that contains the M over Z values of my first scan and the M over Z values of my second scan. And now we're going to do the same thing for the intensities. So I had 1, 2, 3, 4 M over Z values. So I need to provide 1, 2, 3, 4 intensities for my first scan. And here I had 1, 2, 3 M over Z values. So I have to provide 1, 2, 3 intensities for my second scan. And so let me now create a new column that I'm going to call intensity, because that's exactly what these values represent. And now here is how our data looks like. Here on the right-hand side, I had my original data set that now contains 2 additional columns. And there are different ways of specifying this, but here I'm showing kind of the simplest one. An extra column that contains a series of factors representing M over Z values, and another column that represent factors containing the intensities. Okay. So far, there is not much specific to mass spectrometry here. It's simply a data frame that contains information. Well, here is where the Spectra package kicks in, where we can use the Spectra function. And that Spectra function is now going to take that data frame in its input to create a new variable that I call SP here. Sorry, SP. And now SP has used that data frame and has converted it into something that is specific to mass spectrometry data, and that's an object of class Spectra. So Spectra is used to name many things. Spectra is the name of the package. Spectra is the name of the function that creates these objects. And Spectra is also the name of these objects. Okay. And indeed, here it says that I have an MSN data, and because there can be 1, 2, or even more levels, with 2 spectra. And indeed, that's what I created initially, a piece of object or a piece of data that contained only 2 spectra. And this is technical detail. Maybe I'll be able to discuss about it a little bit later. Okay. Any questions so far? So what I think is important here is to see the relation between this code or this output here and this figure. I created a data frame plus information about the M over Z values and the intensities. And using this information, I created an object of class Spectra. And because it's not only a data frame anymore with a set of rows and columns, but it's a Spectra object, now the Spectra package can be used to do some proteomics or mass spectrometry specific data manipulation. Now, if you want, the software R knows that this is mass spectrometry data, and we can now start kind of querying the data in specific ways. So how can we do that? Well, let's use a function called Spectra Variables with SP. And here, Spectra Variables are all the columns that we had in this large data frame, the original data frame that I showed you on the slide. Even though I only provided the MS level and the retention time, because it knows that this is supposed to be a Spectra object, it knows that it should also have other information like the acquisition number, precursor M/Z, precursor charge, and so on. And they are currently missing, as you can see here, and A, right? They are currently missing, but in a typical real-life mass spectrometry experiment, all these data would be available. And these can be accessed. The name of these variables we can access with Spectra Variables, and Spectra data can return that data frame. That is currently mostly composed of missing values, because I didn't hard code these, I didn't type these manually. I only find the retention time and MS levels. So this is exactly what I showed before on my slide here, corresponding to this table. Okay. What you see here in R is exactly what I showed here on this slide. And now we can read the details. We see that we have an MS level. We see that we have a retention time. So these are all the columns in this table. An acquisition number. What was the index of acquisition of that scan? What else? The precursor M/Z value. So if it's an MS2 scan, we need to know what was the M/Z value of the parent ion. What was the intensity of the parent ion? What was the charge of the parent ion? All this information is available as part of this data frame. But mass spectrometry data is also this part with the raw data. And this we can, of course, also access with other accessors, other functions, in particular, peaks data. So if now I run peaks data, it says, well, peaks data is a list of length 2. And let me access the first element. Well, the first element of that list is, as I mentioned before, a matrix with the first column providing information of the M/Z values, and the second column information about intensities. Okay. And so what I would like to do now is to repeat what I have done with a small, with a tiny data set, but do it with a larger real-life data set. Okay, so let me add some, I can share this piece of code afterwards. So let me add some commands. Here, package installation. Okay. And so this section would now be starting with Spectra. Okay, and let's now start a new section, which is getting raw MS data from a public repository. Okay, so let's stop fooling around with this tiny data full of missing values. Let's now try to do this with a real data set. And to be able to allow other people to reproduce this, I'm going to download a data set using a package called RPX that is going to download data from a mass spectrometry repository called Proteomics change. So let me show you first in your browser here. Proteome exchange. There we go. Okay, so it's a Proteome Exchange is a database that aggregates raw data from multiple other repositories where researchers submit their raw mass spectrometry data. And so can I access data here? Okay, so each of these here are unique identifier for some experiments. So this one is called PXD012093. Let me increase the font here. Okay, so these are unique identifiers. And we see where the original data emanates from. And the one that I'm interested in here, it's called PXD and then 501, is kind of the first data set that was submitted as part of testing this Proteome Exchange repository. This is one that I submitted in 2013. It was really a test data set. Okay.
Laurent, is it possible to put the link in the chat, please?
Yes, of course. Yeah. So that's the link to that direct data set. And so let me also provide the link to the general link to Proteome Exchange. And I'm also going to put Proteome Exchange here. Okay. So PXD123451, small data set that we're going to use for the demonstration today. And it's actually a spike-in experiment where we had a yeast background and inside, an Arena background. And inside that Arena background, we have spiked in enolase, BSA, FOSB, cytochrome C. These are kind of human proteins that we have added into this Arena background. And so to download this data from R in a reproducible way, the only thing that we need to know is this identifier, PXD 5 zeros and 1. So let me do this in R. So I need to have the RPX package. And if you don't have the RPX package, you can install it with install from BioC Manager RPX. So it's also a bioconductor package. So that will install RPX and any dependencies. So here I load it. And then I'm going to say, I want to create a Proteome Exchange dataset that has identifier PXD123451. So this function, and I hope this is going to work because currently the servers at the EBI have some issues. But you see, by simply providing the name of the identifier, oh dear. Okay, so let me try to let's cross our fingers. Yeah, so I'm going to copy the output of this in here, just that we have something to see in case I continue to have connection issues to the EBI. But this variable, px1, now knows how to go and send queries to the EBI repository. And it tells us that I have 11 files. Here it gives us the name of the first file and the last file. And if I want to see exactly what kind of files I have, I can use the px file function, and then px1, and then again connection problems. Again, connection problems, but it would give us a list of the 11 files that exist in that data set. I'm really sorry about this, but I think the EBI is migrating their servers, and lately there have been continuous connection issues. But at least you saw that it worked once. And so here are the 11 files that we can access, that we have access to as part of this first test solution. And the one that we are interested in is this one, is the number 8. So the extension that you see here, MZ, XML, oh, sorry, no, I think we'll have the number 7. So the extensions MZML, MZXML, or even .raw here. These are the the file formats for raw mass spectrometry data. And so what we are going to do is to say, well, I want to download this file. So I'm going to store its name simply here in a variable called f. And then I'm going to say px get px1 f to say, well, from all the files that are available in my variable that emulates this Proteomics exchange data set, I want to download the one that is called f here. There would be different ways of doing that. And let me simply store the output here in mzf. And so, of course, I'm being unlucky here. Let me try to run this multiple times. Oh, dear. Of course. Wait, maybe if I disconnect from my VPN. Maybe I'm being lucky here. Can you first confirm that you can still hear me? You can still see my screen?
Yes.
Okay. And so, sorry, I'm going to delete all this because that's very annoying. Let me delete all this. Okay. So pxget is going to check the remote repository and download that file of interest. Now, all these connection problems I had have nothing to do with the size of the file, because the RPX package is clever in the sense that it's not going to download files twice. Every time I download the file, it's going to store it in a central location, a central cache location for RPX. And given that I have already run this code, it knows it. And then instead of downloading the data, it says that it's loading this file from cache. The first time you would run this line of code, it would take, I don't know, maybe 20, 30, or 40 seconds to download that file the very first time. Okay. But now, despite my connection problems, I have this MZML file. MZML is a format for raw mass spectrometry data. And it has been loaded into this directory. And that's the specific cache for the data that were downloaded using RPX on my computer. It would be completely different on your computer. But now I know that I have this raw data file that is stored on my computer. And now I would like to reproduce some of these simple data manipulations that I showed before. I want to first create a Spectra object and then repeat some of these data manipulations on a single but real mass spectrometry experiment. Any questions here? Okay.
Karen is asking a question.
So she's saying, when downloading a public repo, where does one access the biological clinical background to the samples in RPX package?
Yes, that's an excellent question. And so let me come back here. So here, can I not see the files? Well, you know what? Let's take a full experiment list, and let's just take one randomly and see what they provide. So, hematopoietic stem cells. Yeah, let's simply look at this experiment, and then here I can also look at the FTP location. Why don't you want to open? Oh, HTTP. If not, sorry, let me maybe take another one that comes from Pride. Here is this Pride project. Okay, so here I'm kind of navigating to the page of this experiment on Pride. And so here are all the files that are available for that experiment. And so let's see if I display more. Oh, here there is experimental design. No data. Let's see, what do they have here? CSV files. Raw files. So this is raw mass spectrometry data. Raw. Is that all they have? Apparently, yes. So apparently here, or maybe I would need to look at these CSV files, but maybe here it doesn't look like there's a straightforward way to find the annotation of these samples. I have been a little bit unlucky. Let's see if I try another one. Let's try this one. But normally what you would expect, right project here. So you have a kind of a general description, of course, of that comes with the paper. No data for experimental design. Protein groups, no. Checksum, no. And again, final protein list. I don't know. Maybe we would need to look. Maybe there is some information in the final protein list. And I don't know. Have I been unlucky, or is it just that people that submit proteomics data are not very good at providing the experimental data? I don't know. Let's try. Let's try a last example. Pride project. Let's see. Okay, that's really embarrassing. What files do they have? Lots of Excel files. Maybe we could find that information. Next ones. So raw here is all raw mass spectrometry data. No. So ideally, one of these files would be a file that describes the experimental design. And that's that file we could then download. And so if it's a CSV file, for example, that describes all the samples, I don't know, that could be called metadata or sample description, then we could download it with RPX exactly how I did before. So for example, I don't remember what I have here in read.txt, but then I could download that and maybe read it in with read.csv or anything into R to get the sample annotation. But the submission of these data doesn't necessarily expect or require the data to be fully annotated. So that's quite disappointing. Yeah, that's a very good question. Thank you, Karen. But just to give you an idea of what kind of files you might expect to find. So all these are raw mass spectrometry data sets. This is an MZ tab file, is a kind of tabular data with quantitative information. And how come I don't have any identification data here? Okay, you might also get identification data, quantitative data, depending on what the people have submitted. Yeah. So, but that will still require some manual exploration, because apparently I checked a couple of them and none of them seem to provide, at least in an easily accessible or understandable way, information about the samples. But let's carry on here, and let's create a mass spectrometry object of class Spectra containing these data. And that's quite straightforward. We simply reuse the Spectra constructor, and now what we give it as input is the path to one or multiple raw data files. So here I use only one, but I could have here a vector of length 10, 100, and all these files would then be parsed and prepared for bioanalysis. So I'm going to create a new variable called SP. And now Spectra is going to go and read that raw data and parse it to create a Spectra object. So let's see, not sure why this takes. It takes a little bit of time. Okay. I'm sorry, I'm on my laptop here. I initially anticipated to do that on the desktop computer. And it's running Zoom. And I've realized that this slows lots of things down. This takes way longer than it should, but let's be patient. That's not normal. Let's see. I'm looking at the resources that are used by my computer, and I see Zoom uses a lot. Spectra, yeah. Okay. So for whatever reason, it worked here. So let me kill this session. Yes. And move on to this one. Okay. I have no idea what happened here, but we do have our object that is displayed now on the left-hand side. Okay. So SP, that was created with the Spectra function, is an object of class Spectra that now contains 7,534 spectra, which is a very small raw data file. So we expect to see a data frame and then kind of the raw data containing the M over Z values and intensities for the 7,534. So as before, type this in here. You know what? Let me copy the code that I used before, and let's rerun it now, but on the real life data set. So now I have 34 variables in my data frame. Okay. I can access that data with the Spectra data accessor function. So if I do that, I don't see very much. I only see the first row. But I see that for the very first spectrum, it is an MS level 1. It has a retention time 0.45, acquisition number 1, and so on and so forth. So not much of interest here. It's the very first one. So instead of trying to extract the whole data frame, let's maybe try to extract specifically some variables of interest. Oh, and by the way, you can also use length SP to see that we have indeed 5,334 spectra. Okay, but so if we want to see the MS level, well, we can use the MS level accessor, and that will now return the MS level, so 1 or 2 of all our spectra. And then we see here that what is it, 100 first scans that were acquired were all MS1 spectra. So let's feed this into the function table. And here now we see that among these 7,534 MS levels, we have 1,431 MS1 scans and 6,103 MS2 scans. Okay. If we want to look at the retention time, we can do this with the R-time function, SP, and this now returns the retention times of all of our spectra. If we say, well, what's the range of retention times? Well, it starts at half a second, but that's no actual data yet. And the latest scan, the scan that was acquired at the very end, had a retention time 3,601. And Spectra provides a whole set of accessor functions to extract, bits of information of interest. Okay, so we can also access the raw data. Well, actually, first, instead of accessing all the raw data, let me show you some of the filtering functions. So filter MS level is a function that will take all spectra that currently are contained in the spectra object, but only keep the MS2 level spectra. So let's call this SP2, for example. And if I run this, now I have a new object that indeed contains 6,103 spectra, but all of these have MS level 2. Okay, if I, for example, was interested, oh, wait, let's say, chromatogram SP, let's call this CHR1. Oh, sorry. Yeah, I don't need to do that. Let's start by visualizing some of the data, some of the figures that we had before. So let's try to visualize the chromatogram, so the intensities over the retention time. And that's actually quite easy because we have seen here that we can extract the retention time And there is another accessor method, that is the TIC, that gives us the total ion current, so the total intensity for each of each scan. Okay, and so to create the figure that shows the total ion current over retention time, what we are going to do is, because this only really makes sense for MS1 data. So let's extract first the MS1 data. I'm going to call this SP1. And then I'm going to extract the retention time of SP1. Here it is. I'm going to extract the total ion count of SP1. Here it is. And I can now plot this to see the kind of chromatogram. And maybe to have something that makes a little bit more sense, I should say type equals L, to now reproduce the figure that I showed you before. Very straightforward to reproduce. This data only makes sense for MS1 level data. So first, I filter spectra of level 1, and then I extract the retention times, I extract the total ion count, and I feed this into a standard plot function. And maybe something I haven't shown yet is that I can do some subsetting. So let's take the 1000 spectrum. So if I say SP 1000, I will create a new spectra object of length 1. It contains only one spectrum, and that's the spectrum, the 1000 of these, what was it, 7,000 so spectrum. I could also say, give me, create a new object. Create a new object that now contains all the scans from position 1000 to 2000. And here it is. And now I have an MSN object with 1001 spectra, 1000, 1001, 1002, up to 2000. Okay. So the subsetting works like a list. And then in terms of visualization, there is the plot spectra function. So let me take 1000. This is a little bit random. I take spectrum 1000, and here it is. Okay, so plot spectra knows that SP is an object of class spectra and is going to plot it. So it's going to extract the M over Z values and the intensities of this 1000 spectrum. And it's telling us also that it has a retention time of 1106.9 seconds. Note that now that we know what we have in this data set and we know how to access it, we could create that figure manually by, for example, saying here, let's create, I'm going to call this SP1000. I create an object, a spectra object with this single spectrum of interest. I can say MZ of this one. This creates a list of length 1. That's fine. I can extract the first element of this list, the MZ values. I also have intensities of SP123,000. Again, I extract the first element of that list. It's not intensities, it's intensity. Here we go. And so I could simply say, well, plot the M over Z values of that scan of interest, the intensities of that scan of interest, and then say type equals, what do we need to say here? H, to reproduce that visualization, but manually. So the interest of doing it manually is not to produce a figure, but really is to demonstrate the understanding that we have of these data and that we can really access each piece of information one by one. Are there any questions? Because otherwise, what I would like to do now would be to produce this image with you. So I'm first going to explain the image. So at the top figure here, you will probably recognize this figure. It's this third time I'm showing it. It's the chromatogram, the total ion chromatogram of that file that we are analyzing. So we already know how to do that. But instead of saying, okay, here's my complete data set, we're going to kind of focus on one scan of interest, this one, that is highlighted by this red vertical bar. This MS1 scan has been acquired after 30 minutes and 1 second in retention time, and it was acquisition 2107. And here it is. It is this MS1 scan. Among this MS1 scan, 10 peaks were selected to do MS-MS analysis. These 10 peaks are highlighted by vertical bars. And here there is a zoom around the region of the red vertical bar. Okay, so this is kind of a slice of this region from the same spectrum. And then the next thing I want to do is to say, well, I want to visualize these 10 MS2 spectra that were generated by selecting 10 precursor ion in this one MS1, in this specific MS1 scan. So there was a selection at 521, M over Z 521. There was one at 540, and so on. Okay, so this figure kind of encapsulates how mass spectrometry works: separation along the retention time, MS1 acquisitions, and then for a selection of these precursor peaks, of these peptides, selection, fragmentation, analysis, and then acquisition of MS2 scans. So this figure was created with the same file that I showed so far. So what I would like to do now is to really show how I can manipulate the spectra object to regenerate a figure like this. And so I see there's a question here. So the selection of these peaks in MS1 is done automatically by the mass spectrometer. And so one thing that is defined in this type of data-dependent acquisition is the number of peaks that are going to be selected. And so here, this is relatively old data, 10 peaks were selected for MS-MS analysis. More recent instruments that are faster, that can acquire more data per second, you typically have 20 peaks that are selected. And so now the big question is, well, which peaks are selected? It's typically the most intense precursor peaks that are selected. And 20 seems to be a reasonable compromise. We could want to use more, because this information here in MS2 is the information that can be used for identification of data. Okay, to know exactly what peptide sequence this is. And so on one hand side, you might want to say, well, I want to acquire as many MS2 scans as possible for each MS1 scan. However, while the mass spectrometer acquires MS2 scans, peptides still elute from the chromatographic column, right? That the column doesn't stop, the elution of peptides doesn't stop, so that the mass spectrometer can start to acquire these MS2 scans. What happens is that there is a constant, continuous elution of peptides. And so the mass spectrometer quickly acquires MS1 scans, and then it sees, oh, here's an intense peak. This is interesting, so I'm going to get rid of everything else, select it, fragment it, acquire an MS2 scan, and then carry on another MS1 scan and say, oh, here, this one is still intense. And it's going to do that kind of in a continuous way. But so if you spend a lot of time acquiring MS2 data, then you are going to not have the time available to acquire new MS1 scans. Okay, so there needs to be a compromise between spending time in to acquire MS2 and then miss MS1 acquisitions, or have lots of MS1 acquisitions, but then not select enough of these peaks for MS2 acquisitions. And so typically it's a fixed number of scans. In more recent experiments, it would be 20. And it's the most intense peaks that are selected, because those that are too low don't have a good signal to noise ratio. So it's unlikely that if we take something with a very low intensity, it's unlikely that it will generate a clean and nice MS2 spectrum that we will be able to use for the identification of these data.
Laurent, we have...
Yes?
Okay.
Yes, sorry?
It's okay, thank you.
But now if you look carefully at that figure, you should be wondering that some of the most intense peaks, and certainly the 2 or 3 most intense peaks in this MS1 scan here, haven't been selected, right? Here is a very intense peak, here is a very intense peak, here's a very intense peak. These peaks here have not been selected. And so you might wonder why. And the reason is that we don't want to acquire MS2 scans for the same peptide over and over again. Otherwise, we might accumulate lots of identification data for one peptide, but then miss many others. And so what happens, and that's also done automatically, is that whenever a peak is selected, so an MZ value is selected, that MS value is added to an exclusion list for maybe a minute, so that in the kind of the next second, for the next MS1 acquisition, the mass spectrometer will know, do not select that M over Z value again, because we already have some MS2 data. Otherwise, we would waste too much time acquiring MS2 data for the very, very intense, very abundant peptides. And we would never acquire data, MS2 data, for the peptides that have a low intensity. But what we want is a good coverage. And so the reason why these intense peaks aren't selected is most likely that they were acquired during a previous MS1 acquisition and thus were added to the exclusion list for a certain amount of time. Okay, so let's get our data and let's try to reproduce all these visualizations to kind of demonstrate that we indeed are in a position to manipulate, first understand and manipulate these data. Okay, so let me add here visualization Okay, and the first part would be a chromatogram. So I have already, where was the code? I'm going first to copy that piece of code here. So this is the piece of code that I used to produce the chromatogram. But I'm going to use it differently now to illustrate the kind of flexibility. Oh, let me first start by producing the figure like this, and then add the red bar, okay, to exactly reproduce the figure that we want to do, to add the red bar at the very specific retention time of my 2807 spectrum. And so we know that we can extract the retention time. Okay, so here they are, all of the retention times, but the retention time I'm interested in is 2807. So that's the retention time of the MS1 scan that we would like to visualize. And so let's visualize this like this. And why? Oh, sorry. I need to say that this should be a vertical bar. So here it is. Okay, and that exactly reproduces my figure of interest. So let me show it again. Here, top. Here is the figure retention time. And I have a scan of interest that is marked by this vertical red line. But there is actually another way to manipulate these data, and that would be by using the pipe. So let me first load, let me load the whole tidyverse. Okay. And what I'm interested here is, the first part of the tidyverse is that I will be able to say, to use the pipe argument. Okay. So for example, here, I take my object that contains all the data, and I'm going to pipe it into the filter MS level function to only keep MS1 level data. By the way, is everybody familiar with the pipe? So it's the classical pipe of tidyverse. Please let me know in the chat if you would like me to explain this syntax that is equivalent to this here. Because I'm going to use the pipe multiple times here. Okay, so I take my full data set and I pipe it into the MS level function to extract MS1 level data. And now I'm going to say spectra data, because what I would like to do is to extract this big data frame that contains all the information. But as you can see here, it's a data frame, but with a capital D. So it's a kind of bioconductor-specific data frame. But what I would like to do is to convert it as Tibble. Okay, so here it is. It contains exactly the same data, 1,431 rows, one for each MS1 spectrum, and here 34 variables, MS1 level retention time. And now I'm going to pipe this into ggplot. And I'm going to say that along my x-axis, I want retention time. Along my y-axis, I want, let me check, total ion current. Here it is. Okay, plus geom ion. And so with the flexibility that the Spectra interface offers, I can fall back to the tidyverse tools and tidyverse way of handling and managing data if I want to. So I can do it manually, step by step with the plot function, or I could, for example, use the pipe, extract my data, convert it as a tibble, and then use ggplot to visualize that chromatograph. So whether you prefer one or the other is really a matter of taste. But well, here we go. If you like to like piping and like ggplot to produce your figures, you could quite easily do that once you're familiar with the functions, the different accessors and extractors. So that's, but let me switch back here. That's for the chromatogram. Let's now extract the set of scans that are related to this scan that is highlighted here in red. So what was it, 2807? Okay, so it's this object, sorry, this spectrum. So it's an MS1 level. And actually, let me just do a few things manually here. 2810. Okay, so actually what happens here is that my spectrum of interest, 2807, is an MS1. And then right after that one, I have a set of MS2 scans. So let's, okay, let's, sorry, yes, 18. Okay, so look, if now I extract scans from 2807, which contains my spectrum of interest, and then 10 additional ones or 11 additional ones, I have here all the MS2 scans that originate from this one, and then the next MS1 spectrum. Okay, so what I will need for my visualization, and let me remind you of my visualization here, I will need this one to produce this first figure, and then I might want to do some zooming. And then I will also need all the other ones, the MS2 scans that follow this MS1 scan, to produce the MS2 scans whose precursor peaks come from this MS1 scan. So here I have done it manually, so to speak. I know that this is the index of my MS1, and then here I counted kind of 11 more just to see the following MS1 scan. But there is a function to do that automatically, and that's called filter precursor scan, SP, and then I provide an index of interest, filter precursor. Of course, it doesn't work with the typo. And this function will automatically go and check in SP, look at the index 2807, and automatically also extract all the scans that originate from this one. Okay, so by doing this, and I don't know, let me call this SPX. By doing this, I now have the subset of 11 spectra or scans out of the 7,000 and something. And these 11 scans are those that I will want to visualize. So let's start easy. Plot spectra, SPX, and the first of these, that is here. So this is the MS1 scan that is also shown here. The only thing is that here I cut the x-axis just after 1,000, so just after here, but this is the complete data set. Okay, so with plot spectra, it's quite easy to regenerate a figure. We could also do it manually if we wanted, but let's use plot spectra. But what we want to do now is to highlight the peaks that were selected for MS2. And this is something, that's the information that we have with the, what's that? Oh, I did something with my, okay. Does anyone know? I suppose that's something from Zoom. Does anyone have an idea how I can get rid of this?
Oh my God, I think. Did you paint this or?
I have no idea.
Because we had this once when we've been hacked, so.
Ah, okay.
So, hello.
Yes.
Yes, do you need to check maybe the list of participants here? And
I mean, I don't mind. We can carry on with this blue line.
okay.
Yeah, let's try to see if we can carry
Let's see,
on.
wait, can I not clear? Clear, clear all drawings. Okay. Or maybe I clicked something I wasn't supposed to click.
Okay.
Okay.
Fine.
Because we had this bad experience once and
Okay. Well, let's see. Let's see if so where was I? Yes. I produced this visualization of my MS1 scan, and I would like to be able to pinpoint those peaks that were selected for MS2. And that's quite easy because I can, among this large data frame I can extract here precursor MZ of SPX, the M over Z values of the precursors. Okay? And that's what you see here on the left-hand side. So SPX contains 11 scans. So I have 11 precursor scan values. So 11 M over Z values, except for the first one, because the first one, if you remember, MS level SPX, the first one is an MS1 scan. So there is no precursor. All the other ones are MS2 scans. And here I now exactly have the precursor values, the precursor M over Z values, or the M over Z values of the precursors. And so after plotting the figure, I can simply say AB line, I want to add vertical lines using, well, the precursor M over Z values. So here I'm simply going, just to keep it clean, I'm removing the first one, DNA, and here, oh, and let me change the color. All equals gray. And here we are. And you know what? Let me also here, I think I can say X lim equals C from 100 to 1000 should work. 100 is too. 400. And here we go. So now I have regenerated that figure that shows the raw data for my MS1 scan of interest and the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 precursor precursor peaks that were selected for MS2. And so the only difference is that one was highlighted in red. Let me show you the figure here. One was highlighted in red, and I think it was simply the first one, but that's not very important. I could add this here. So the first MS1 scan, which would be the second precursor MOZ. So here it is, but that's only a little detail. And so here we have seen that we can already zoom in. And so if I look at the M over Z values, right, okay, of my first one is 521. So now if I would like to zoom in more precisely on this M over Z range from my first acquisition, well, I could say I want to zoom from 521 to, for example, 522. And here it is. Okay, so here we have the different isotopic peaks. And actually, maybe here I should say type equals L. This is more relevant for this type of visualization. So here we have a detailed zoom in on this precursor, this peak that is going to be selected for MS1. And so to kind of finish this visualization, I'm going to redraw this one here. I would like to visualize all the MS2 scans. And here I know that all my MS2 scans are actually SPX from 2 to 11. Or actually, let's use some of the functions that we already know. Okay. So this will now extract all the MS2 scans. And now if I say plot spectra, it will automatically produce the 10 figures for these 10 MS2 scans. Okay. So let me take the figure here that we wanted to initially produce, and let's walk through the code. Here we go. So the first dimension here was the retention time, and we can easily access it with the RT accessor. But given that this is only relevant for MS1, we filter only MS1 scans, we extract the retention time, the total ion count, and we simply feed this into a plot function, or we can use ggplot to produce this figure. Now we want to focus on a set of scans of interest, including one MS1 scan here and a selection of the set of MS2 scans that originate from that MS1 acquisition. We can simply use filter precursor scan for that, where we provide the complete data set and then the acquisition number of a spectrum of interest. And that will automatically create a subset that contains the MS1 and the 10 MS2 scans. And then visualizing scans is also trivial with plot spectra, although it would be also very easy for us to either do it manually with the base plotting or using ggplot2 if we wanted. But we simply extract the MS1 scan to visualize it, or we extract the MS2 here scans, and we visualize them also with plot spectra. And then if we want to identify the M over Z values of the precursors, we can use the precursor MZ function to extract exactly that information from our raw experiment. And so everything that I have shown really leads from the understanding of this data structure, where we have a data frame that contains all sorts of variables, metadata that describes these data. Now, you can't read what is written here, but we have seen some of these variables, and we have used some of these variables. We have MS level, total ion current, precursor M over Z, retention time, and so on. And all of these can be accessed with specific accessor function, RT, R time for retention time, precursor M over Z for the precursor M over Z, and so on and so forth. And this is, for example, to produce the first figure, the chromatogram here, we only used data from this table. We used one column that provided the retention time and another column that provided the total ion current, as shown here. To visualize or to access the other data, that was the raw data, so M over Z values and intensities. So here we want to access these data of interest here. Well, for that, what we did was filtering based on an information in the main data frame. And then we use the plot spectra that automatically knows to extract the information in this part of the data. We could also have done it manually, as was shown here. Right, we can use MZ and we can use intensity to extract these parts of the raw data manually. And so producing this figure, of course, does not represent a full analysis of a mass spectrometry experiment, but I think it is a good illustration of what Spectra in particular allows you to do when, on the one hand side, you understand the data structure, okay? This is understanding the data structure. And on the other hand side, you know how to make use of the data structure to access the different parts of interest. And I think that gives you a nice set of tools to be able to explore and really understand mass spectrometry data beyond kind of feeding them into a black box, which is also something that you might want to do if you want to use the specific software that will kind of, analyze, quantify your data. But in some circumstances, you might want to have more fine-grained control and understanding of your data. Okay. Any questions here?
So, don't think there is a question.
Sorry, where is the question?
Oh. Yes, so you get a question from
yes.
Karen.
How does one assign identity?
Yeah.
Yes. So for that, I'm going to move to another chapter, which is identification data. So this tutorial kind of gives an introduction, shows how to analyze and manipulate raw data, which is what I showed you. And then there is a chapter on identification data. There is a chapter on quantitative data. So let me start by explaining. I thought I have a figure here. Let me start by explaining how identification works and how these MS2 scans are used for identification. So what we have here is a peptide composed of 4 amino acids. So the first amino acid here is residue number 1, second amino acid, residue number 2, residue number 3, residue number 4. So this is the chemical structure of a peptide with an N-terminal and a C-terminal part. Now, what happens when we dissociate, we break up that peptides? What happens when we fragment that peptide? Well, that peptide can break between any residues here. So it could break between residue 1 and residue 2. And that would generate one bit that contains only residue 1, and then the rest of the peptide, the C-terminal side of the peptide that contains residue 2, 3, or 4. Another peptide, because we are analyzing thousands of peptides here, you know, this one peak is not a single peptide. It's many copies of that peptide. Another one might break between residue 2 and residue 3, right here. That fragmentation would produce an N-terminal part containing residue 1 and residue 2, and a C-terminal part containing residue 3 and residue 4. And then there is a third position when they can break to produce residue 1 to 3 on one side and only residue 4 on the other side. Now, if we look more closely, where exactly this fragmentation can happen, it can happen between the first carbon, second carbon. It could happen between here C and N, and it could happen between N and C. So when we fragment this peptide to produce N-terminal R1 and then C-terminal R2, R3, R4, we can actually either have a fragmentation along this bond, and then we talk of an A ion and an X ion. It could fragment exactly here, and then this would be called the B ion, and the rest here on the right-hand side, the C-terminal part would be called the Y ion, or the fragmentation could happen here, and then we would have the C, or in this case, C1 ion, and then on the right-hand side, the C-terminal would be the Z3 peak. Okay? And so these are all the fragmentation patterns that we can get for a peptide of length 4. Now, each of these A, B, C, and X, Y, Z ions, and actually we generally only see some small A ions, but mostly B and Y ions. Okay, so let's only focus now on the B and Y ions. Well, these B and Y ions have a specific mass. So depending on the actual residue, the amino acid here in position R1, we know that B1, we could calculate the specific mass of B1, and we could calculate the specific mass of Y3. Similarly, if it breaks here, we have here B2, and we can calculate the specific mass of B2 if we know R1 and R2, and same thing on the C-terminal, the Y2, we can calculate the mass based on R3 and R4. Okay? Well, this now should remind you what we see here. So MS1 scan, MS2 scans, this is the fragmentation pattern of the one that is highlighted in red. This is the fragmentation pattern of the one that is highlighted in orange. What are these peaks here? Well, these are the fragment peaks. So this is a collection of fragment peaks, B ions, Y ions. We also have some of the we have the A, B, C, and X, Y, Z ions, but generally it's mostly the B and Y ions that we can detect with kind of time of mass spectrometry that we use. And so I have some visits. So identifying a peptide essentially means being able to match the observed fragment peaks, okay, those that we can read here, and theoretical or expected, and here expected fragment peaks. So if I am able to match, and so where do these theoretical fragment peaks come from? They come from a database. Okay, so if I'm analyzing a human sample, I will also download from UniProt which is the kind of a very important database that contains information about proteins, there I can download a FASTA file with all the expected protein sequences. And so I'm going to instruct my computer to do the same thing that I did in my tube. So in my tube, I use trypsin to cut my proteins into peptides. And these are the peptides that I will analyze by mass spectrometry. So I can use, in my computer, I could say, use this protein database and cut it just as the trypsin enzyme would cut it. I'm going to show you a bioconductor package that you can use to do that. And it's called cleave-R. There we go. And let's look at the vignette. So every bioconductor package comes with a vignette, okay, which is a kind of a lengthy documentation that describes the package. Well, the cleave-R package here, for example, if we take this small amino acid and we say, well, cut it with the enzyme trypsin, well, trypsin is going to cut this peptide into two smaller peptides, this one and that one. Okay, and we can do that. I think there is an example with a bigger example here. So here, for example, this code, and we're not going to go through this code in detail, but it's going to download two protein sequences that have these two identifiers. Okay, it's insulin and somatostatin. These are the two identifiers. These are UniProt identifiers. So here we have the sequence of these two proteins, and we're going to say, let's cut these protein sequences using another enzyme, that is pepsin. And that one will create these possible peptides. Right? So if we had a very simple biological sample that only contain these two proteins, then these are all the possible peptides that we would observe in our sample. Now, many of these are too small, MA, LL. All this is way too small to be analyzed by mass spectrometry. However, generally we consider peptide length of 6, 7, and beyond to be long enough to be able to be, so that we can confidently acquire. If we knew that we had a very simple sample containing only these two proteins, these are the peptides that we could observe. We ignore the tiny ones, but all those that have a length greater than 7 amino acids are those that we could potentially observe. Okay, and so if we take this peptide, or let me move back to my other presentation. Here, if I have this peptide sequence, so here I'm again working with a SP object that contains raw data, and among the raw data, or the annotation data, I also have sequences. I have this peptide. Well, if this is a peptide that I expect to see, I can calculate the M/Z values for all my possible fragment ions, like the B1, that contains only T, the B2, TH, B3, right, the 3 first amino acids, and so on. So the Y1 here, the Y2 here, and so on. And so this table is much longer. And so these are all the expected M/Z values for the fragment ions. These are the expected M/Z values for the fragment ions. And here, for example, we have the observed M/Z values for MS2 scans. And so what is identification? Identification is comparing a set of expected M/Z values for fragment ions. And where do these come from? Well, these would come from all possible peptides that we will expect to see for a specific sample of interest. And then all the observed M over Z values, well, these are the M over Z values of all the MS2 scans that were acquired during the experiment. And so I think I have a visualization here, for example. And this is also something that you can reproduce yourselves. Here, we use plot spectra to generate a figure exactly as we did before. But in addition to that, for this peptide, we have a sequence. And so we ask to add the fragments. This is a function that will be add the fragments. And here, for example, so this is kind of outstanding to find as many fragment ions. But here, as you can see, we have the B1, the Y1, the B2, B3, Y3, Y4. So we really have the full series of Y ions. We have quite a few, or maybe also the full, or not quite the full series, but we also have many B ions. So this is exactly what it means to identify a peptide. It means matching observed fragment peaks that were acquired during mass spectrometry and expected fragment peaks that can be calculated like this. And this uses the calculate fragments function from another package called PSM. So let's try to run it ourselves. So if I remember well, it's in the package called PSM. No package PSM. I recently updated my R version, so that's fine. Let me install it myself. So I'm going to install. Yeah, and that's good because it allows me to show you. So the package PSM is not yet available on Bioconductor. It is only available on the for mass spectrometry.org. Okay, so there is a list of packages, and here it is. So it's currently only available on as part of the R for mass spectrometry GitHub page. Okay, but it's still it does come with all sorts of documentation. And so if I want to install this package that does not yet exist on a Bioconductor, then I need to specify R for mass spectrometry here, and then the name of the package. And this will now go and download the PSM package, not from the Bioconductor repository, but from GitHub, the PSM repository in the GitHub R for mass spectrometry organization. And I don't want to do any updates now. Okay, so installation of BioC Manager, installation of packages that are already on Bioconductor like this. If one of the R for Proteomics packages is not yet available on Bioconductor, then you would need to specify here R for mass spectrometry so that this function automatically installs it from GitHub. Okay, so library PSM, and then I can here say calculate frag. Try. Yeah, here we go. So calculate fragments, and then it takes as input a peptide sequence, and it returns a data frame with, you see, all the B and Y ions that are possible to obtain with this sequence. Of course, this is not something that we do manually, right? So here, if I think it's $MZ. Yes, so these are all the possible M over Z values that I can obtain for this peptide. And so I would try to match these M over Z values to all the scans in my acquisition. So, of course, this is not something that we do manually. There exist dedicated pieces of software that do that. These are called search engines. And let me type some of these names here. So there is MSGF Plus. That's a freely available. What else do we have? We have something called Comet. We have mascot is a commercial one that is widely used. Anyway, there are metamorphers. Anyway, there are many, many more, and too many to cite here. But so we would use these dedicated pieces of software that are going to do exactly that. Do all the possible comparisons, or maybe not all comparisons, but possible many, many comparisons between expected M over Z values for a peptide that is a human peptide if we analyze a human sample, and then all the M over Z values that are produced, that are acquired by mass spectrometry. So this is how the identification works. And this is something that is done on an within an external software. And I can actually show you how to, so let me add a new section here. Identification. Okay. So I have a package called MS, there is a package called MS Data. And again, if you want to install it, it's on Bioconductor. So you would say MS Data. And here is an identification file. And actually, I can also show you Proteomics, I think. Yes. So we also provide example data files as part of, we disseminate these in the MS Data package. And so here in ident, the ident function from MS Data will return the name of this one identification file that I'm going to use here. I could also download it, I believe, from Proteomics Change. But here I'm going to use the one that is already installed. And then if I say folder names equals true. Okay, here it is. And so I can now read that data in PSM, the PSM function, if I remember well. Let me call this ID. And, sorry, ID. And so in this identification file, okay, so that's, sorry. Okay, of this identification file, and the identification files have generally the extension MZID, right? MZML for the raw data and MZID for the identification data. And there exist several different types of format, and I'm not going to get into this. But here the MZID is a dedicated type of file that can be exported by these search engines. And if we use the PSM function, that PSM function is going to parse that MZID file and return an object of class PSM. So again, here we have PSM for the name of the package, for the function that reads the data, and for the class of the output. And that data set now contains identification data for 5,802 matches. So PSM stands for peptide spectrum match. So that's when there is a match between an observed, acquired spectrum and possible. I see there's a question. Yes.
Yes, we have a question. So.
Okay, so we don't take every permutation. We take all possible peptides. We take all possible peptides in a protein. So where was that example? So here, these two proteins, if we cut it with the pepsin enzyme, typically we use trypsin. You know what? Let me run this code. So I don't think I have this. So IOC manager, let's install. I'm going to copy this at the top with all the installations, and then library. And in the meantime, okay. Don't want to update. So I load, so the UniProt WS is for web service. So that's a package that allows to send queries to UniProt. And so here the tax ID is for human. And so this function is going to initialize a connection to the UniProt web services. And so this might take a little bit of time. And then once this has been done, saying that I'm interested in pure proteins from the taxon 9606, which is human, I should then be able do I again have connection problems? Let's see. So here it okay, done. So I have created this selection, and now I'm going to download the peptide sequences. Okay, so sorry, the protein sequences. So here, this is the sequence from first protein, and this is the sequence from the second protein. And so there is a little bit of data wrangling here. I'm going to go over this very quickly. What we get is kind of a vector of length 2. And there are more efficient ways of doing this with using, I don't know if you have heard of the biostrings package, which is a package that can allow, that allows you to handle and manipulate kind of genome size sequences. But so here we have first protein, second protein. And what I'm going to do here is that I'm going to say trypsin. And of course, I need to load library cleaver, which I have. Very good. And so here it is. So assuming I'm only interested in these two proteins, the possible observations, the possible peptides for these two proteins, assuming I use trypsins, are all these peptides. And so I could then say, I don't know, let's take this one. Okay, so calculate fragments for this peptide. Well, if this peptide is present in my sample, then I expect to see $MZ. Then I expect to see an MS2 scan that has, maybe not all, but a certain proportion of fragment peaks that have a match to this one. And if there is a good match, that good match we call PSM. And so it's not kind of all permutations, it's all possible peptides. But yes, feel free to ask it directly if it's easier.
Yeah, maybe it's better if I unmute Karen.
Hi, can you hear me all right?
Yes.
Okay, perfect. Sorry, I wasn't quite following. So just to summarize, to make sure I've understood it correctly, there's two components we're trying to bring together in order to assign identity. So the first component is for our sample of interest, which has X unknown proteins And whenever it goes through mass spec, it's going to be fragmented essentially by brute force in a stochastic manner.
Yes.
And then at the end of the analysis bit, we have the M to Z ratios with the different intensities.
Yes.
Then,
sorry, I want to clarify one thing, is that what enters the mass spectrometer are peptides. These are not proteins. We cut the proteins into peptides in the lab before starting the acquisition, but otherwise, that is correct.
Oh, right. So there is enzymatic pre-digestion step. All right. Okay. Sorry, that's what I misunderstood. So then we then combine. So then the second component is empirically derived enzymatic fragmentations from known proteins, which then gives known peptide fragments. Okay, and there's homology between the enzyme that we use for digestion in our sample versus what's publicly known. And that's generally trypsin, is that correct?
Yes, most experiments use trypsin because when you use trypsin, it doesn't cut too much, right? Otherwise, we get too many very short peptides, and that's not good for mass spectrometry. If we have an enzyme that doesn't cut enough, then the peptides might be too long. So trypsin is the usual suspect that is used in most experiments.
Okay, so this essentially becomes like a probabilistic mapping problem, which still is probably computationally very expensive.
Yes, it is computationally expensive, although some of these software are very fast. Some of the more recent software are extremely fast, so that's not too much of an issue. But the problem is not really computational, but it's more statistical, right? So so we can assign a score, right, depending on how many matches we have between the scans, so the raw data, and the expected data. We can calculate the score. Let's say that score is simply the number of matches. It can be something more complex, but that doesn't matter. But so how many, or what's the percentage, or what's the score that you need to be able to confidently say, yes, that peptide was present in my sample? And that's a question that is quite difficult to answer. But we need to answer it to be able to say, this is a peptide that we believe was in the sample. And so how does that work? And that reflects, that I think identifies why it is difficult to identify peptides. And normally I have, maybe. Okay, I do have, I can produce that figure here. So I have my ID. Let's just say as Thibault, this identification data. And among this identification data, I have scores. Here, MSGF raw score. Okay, that's a quantity that will tell me how good do I have a match. The higher the score, the better the match. But it's still extremely difficult to specify, to say, that's a score of 20 is good, or and when we believe it, or is it 20, is it 5, is it 10? We need to have a kind of a more statistical understanding of how good the score is. And so what happens? So if I look in this table, there is a column that says, is decoy? It's a logical. And it tells me whether this PSM, so this result that I have with a specific score, has been obtained by matching a spectrum to the actual, so let me, to an, sorry, no, this is what I wanted to show, to an actual peptide that we expect to see, or to what we call a decoy peptide. And the decoy peptide would be the reverse of that peptide, or actually the reverse of a protein. So we take all our protein sequences that are called here sequences. And then we are going to do the exact same procedure with the reverse of protein, right? Instead of saying, well, this protein has this sequence, we're going to say, oh no, this protein starts with N, C, Y, N, E, and so on. So this reverse database, also called decoy database, is a database that looks like human proteins, but these are not human proteins. And so what we're going to do is that we're going to do this search procedure on the real database, these ones, and on the reverse database. And now we have two distributions of scores. One distribution from the real proteins or real peptides, and one distribution of scores that we know are not correct, right? That's kind of a null distribution of scores. It's a distribution of scores against peptides that are not human, but they are very much like human peptides. And so let me produce that figure here. So I'm going to say here, tibble. So I'm transforming this ID object into a tibble. And then I'm going to say ggplot2. And I'm going to say x equals MSG, MSGF, here it is. So let me take the raw score, for example. Okay. And then I'm going to say geom density. And actually here, color equals is decoy. So what do I have here? And that's a very ugly result. And I have the distribution of scores. So the red ones are the correct peptides. And some of these have very high scores, around 100 and 200. But then, if I look at the distribution of scores of peptides that are blue here, true, they are from the decoy, some of these also have very high scores. And so by using these two distributions, I can calculate a false discovery rate. For every score threshold, I can calculate how many peptides, or sorry, how many PSMs have a score greater than my threshold. And then among these, how many come from my real database and how many come from my decoy database. And by dividing the number of peptides that come from my real database divided by, the number of PSMs that come from my real database divided by the number of PSMs that have a score greater or equal to the threshold, but that come from my real and my decoy database, I can estimate what is the false discovery rate amongst these identifications. And so this is not a nice figure at all, because here the score distributions overlap. They are very, very similar. So I have to admit, I thought that the presentation was tomorrow. And so I would have been in my office with another computer, and I would have used another data set where there we would have seen that indeed the distribution of the red one here would have been shifted towards the right. And then we can say, well, at this score threshold, we know that we expect to see 1% of false identifications that come from the decoy. And after that, of course, we get rid of all the identifications from the decoy, and then we keep those from the real database, and we consider them trustworthy identifications.
Laurent, we have a question from, I will unmute Amma.
Hello.
Yes, hello.
Hello.
Hi, thank you very much. So I didn't join this early, but I have a follow-up question from what Karen asked. So I wanted to find out, I'm trying to do some work with regards to treating some cells with a certain drug, and then to look at the proteome of the parasite after treatment. And I'm supposed to go in blindly, so look at the proteome. So I do not know if I'm going to get any novel proteins or not. So how am I able to identify if after, say, trypsin digestion, the individual fragments and how to put them together?
Yeah. So the identification will work exactly as I say, but instead of working on, so the example that I give here was a very small one, so that we can do it manually, with 2 proteins, and the software is going to do that automatically for you. This will be done on all the proteins of the species of interest. So if these were human cells, you would use the human database, so a database with all the human proteins. And the software will automatically do all these analyses for you to then return a list of proteins that were present in your respective samples. Okay. So that will get you a list of proteins. Something else that is done with this raw data, let me show here another chapter about quantitative data. That is the quantitation of data, of these peptides that you have in your sample. And I'm not going to start this because there are different ways of doing quantitation in mass spectrometry. You can have label-free quantitation. You can have label quantitation where you mix samples together. And this quantitation can happen using, or can be done on MS1 data, or it can be done on MS2 data. But now you know what MS1 and MS2 is. So you have many types of software that will kind of integrate all these analyses for you and will return a matrix, kind of a table with proteins along the rows and samples along the columns. And after that, you would use that table to do some statistical, or there might be some data processing, normalization, and then do the statistical analysis to eventually identify which proteins are present, absent, or differentially abundant.
Okay, so if I get you rightly, it means that it doesn't just work for proteins I'm familiar with, but other proteins I do not know of.
Absolutely. So let me show you here. You would go on uniprot.org. So proteomes here. So here we have some reference proteomes. And of course, I always forget exactly how to do it. Maybe downloads. Anyway, you can download. Oh, I can show you actually. I surely have one somewhere here. Sorry, let me. Okay, I think. Okay, so if I open PXD, so you see PXD, this is something that we have seen. So PXD is a data set for proteome exchange. And this is actually a massive data set that was published a couple of years ago that I downloaded and I wanted to reanalyze or kind of to re-explore. And here is the FASTA file with all the expected, well-known and well-characterized human proteins. So it tells me that it's a large file. Yes, I don't mind. So here it is. So this is a file that I downloaded from Uniprot, where here I have the identifier and a description of the protein. And here I have its sequence. And I have this for how many? Let me tell you how many proteins I have here. So I say grab this, and how was it called? Yeah. Mm-hmm. Yeah. Okay, 20,381 proteins. So this file that was used as a reference proteome contained just over 21 or 20,000 proteins. So of course, you will not identify peptides that are completely unknown. What you can only identify something that was provided with the database. But you don't need to know exactly what to expect. So you can use here 20,000 proteins, and the software will tell you, given that data set and given that database that you provided, I have observed these peptides and these proteins.
Okay, so what happens then if you have a protein that has never been identified?
Yes. Wow. So you will not be able, there is a large fraction of your peptides that will not be identified with the procedure that I just described. Let me maybe stop my, stop sharing. I'm not very familiar with Zoom. So a whole set of scans, MS2 scans, will not be identified for many, many different reasons, but one of them could be that there is a protein that we don't know of yet. Now, one thing that is possible is to do de novo sequencing of these scans. That means we take an MS2 spectrum, and then we measure the distance between the fragment peaks. And the distance between fragment peaks should be the mass, should correspond to the mass of an amino acid. And if you have good quality scans, you should be able to sequence, to infer the sequence without any reference database. But that requires very good spectra. And so there is software that will do de novo sequencing for you. But of course, if you don't know the protein exists, it is quite tricky to identify it.
Okay, thank you very much.
You're welcome. And so to wrap up, because it's nearly 8, and I think we are expected to finish at 8. Well, actually, let me share my screen here. So there are different ways of quantifying that mass spectrometry data. And certainly if you're considering generating mass spectrometry data, this would be a really important discussion to have with the core facility or your collaborators as to what kind of quantitation technology to use. So I have spent a lot of time in talking about how does mass spectrometry work. These were the first chapter here. What is raw mass spectrometry data? I have briefly shown you that you can read this identification data, and I haven't demonstrated, but you can, of course, combine the raw data and the identification data, and all this is described in this online book. There is no print version, and it's still being updated regularly. One thing that I haven't discussed at all, and I think I talked with Hedia to maybe give a second seminar, I don't know when, but whenever there is time that fits, we could also consider a second session where there we would focus on the statistical analysis of proteomics data, right? At some point, you will get a matrix with quantitative values for peptides and samples. And once you have this, there are lots of decisions to be made as how to process that quantitative data. And so that's one part of proteomics that I haven't talked about at all. I have focused on understanding how mass spectrometry works and how can you kind of test your understanding by manipulating raw data. But if there is an interest, we could walk through this material on quantitative data and more specifically on how to manipulate these and how to do a quantitative proteomics data processing. And that would involve completely different packages. So if there is interest, you can all, of course, have a look at the chapter here and the package. The most specific package I'm referring to is a package called Q features. And again, it comes with ample documentation. But if there is interest, we could follow up on that. It would be a completely different type of presentation.
Thank you very much, Laurent, for this amazing workshop. So even Karen shows she's saying thank you for the excellent workshop and the clear discussion of the pipeline and exploration of the statistical aspects would be very helpful.
Okay,
So
well, we'll make plans.
yeah, I think
for the future.
Yeah, maybe we'll have another workshop maybe later. Thank you everyone for joining. I don't know if you have more questions because it's like it's already time. So is there a last question we can ask, or you really want to ask? Okay. So everyone is looking for the subsequent ones. So thank you very much.
Okay, then we'll organize that in a couple of months or a couple of weeks.
Thank you.
Thank you very much.
I see no questions. So again, thank you very much, everyone. And Laurent, thank you very much for this amazing workshop, for attending our workshop, and Laurent for giving us 3 hours of his time explaining all this notion of proteomics and introducing us to the world of proteomics. So thank you very much again.
Thank you.
Let's have we have like a workshop survey. So I launched the poll. So if you can respond to this question. So what do you think about our workshop? So it will be helpful for us, to schedule the next workshop. So.
All right, but I'll be going. Thank you very much, everybody.
Yeah,
and
thank you, Laurent. Thank you very much.
Take care. See you soon.
Thank you. So we have, before going, so everyone is saying that they learned a lot during this workshop, and it was a great workshop. So thank you very much.
Take care. Bye-bye.
Thank you. Bye-bye. Thank you. I will share the result of the. So could you see the results?
Yes.
Yeah. Thank you. Thank you. Okay, so we'll close this session. And thank you again for attending. Monet, do you have something to add before?
Thank you all for attending. Sorry for being unmute later. I'm really sorry. Earlier, I mean. Follow our meetup group, so this way you will be updated with meaning with our next meetups. And that's all. Thank you for being part of our new community. Goodbye.
And if you have, please, if there is like a topic you're interested in, please don't hesitate to let us know. And we will look for a speaker, and we will organize a workshop. Okay, and we will send you all the video and the teaching materials, by May. So, okay, thank you very much.